In this video, we will learn cortical neuron laminar specification and mechanisms of its formation. The cerebral cortex is the outer covering of the surfaces of the cerebral hemispheres. In the human brain, it is between 2 and 4 millimeters thick and makes up 40% of the brain's mass. 90% of the cerebral cortex is the 6-layer neocortex, with the other 10% made up of allocortex. The 6-layer neocortex is a uniquely mammalian structure, and it reaches its most extensive elaboration in humans, endowing us with remarkable cognitive abilities. The neocortex is formed of 6 layers, numbered 1 to 6, from the outermost layer 1, near to the P matter, to the innermost layer 6, near to the underlying Y matter. Each cortical layer has a characteristic distribution of different neurons and their connections with other cortical and subcortical regions. There are direct connections between different cortical areas and indirect connections via the thalamus. Layer 1 is called the molecular layer, 2 is the external granular layer, 3 is the external pyramidal layer, 4 is the internal granular layer, 5 is the internal pyramidal layer, and 6 the multiform layer. Layer 1, the molecular layer, contains few scattered neurons, including GABAergic root-skip neurons. During development, Cajal radial cells and subpale granular layer cells are present in this layer. Layer 1 across the cerebral cortex mantle receives substantial input from matrix thalamus cells. Layer 2, the external granular layer, contains small pyramidal neurons and numerous stellate neurons. Layer 3, the external pyramidal layer, contains predominantly small and medium-sized pyramidal neurons, as well as non-pyramidal neurons with vertically oriented intracortical axons. Layers 1 through 3 are the main targets of interhemispheric cortical efferents, and Layer 3 is the principal source of cortical efferents. Layer 4, the internal granular layer, contains different types of stellate and pyramidal cells, and is the main target of thalamocortical efferents from thalamus core type neurons as well as intrahemispheric cortical efferents. Layer 5, the internal pyramidal layer, contains large pyramidal neurons. Axons from this leave the cortex and connect with subcortical structures, including the basal ganglia. In the primary motor cortex of the frontal lobe, these axons travel through the internal capsule, the brainstem, and the spinal cord, forming the cortical spinal tract, which is the main pathway for voluntary motor control. Layer 6, the polymorphic or multiform layer, contains fewer large pyramidal neurons and many small spindle-like pyramidal and multiform neurons. Layer 6 sends efferent fibers to the thalamus, establishing a very precise reciprocal interconnection between the cortex and the thalamus. That is, layer 6 neurons from one cortical column connect with thalamus neurons that provide input to the same cortical column. The 6-layered organization is central to cortical function and is highly conserved in all mammals despite the fact that the cortex has grown in complexity throughout the recent mammalian evolution. This leads us to question the developmental mechanisms involved in. Cortical progenitors generate radial clones that consist of neurons arranged in multiple layers. Interestingly, a prominent feature of this process is time dependence. That is, cortical progenitors generate different layers of neurons at distinct developmental stages. After the birth and migration of the Cajal radial cells in layer 1 and the subplate cells, the next neurons to be generated in the cortex are the pyramidal neurons of the deep layers 5 and 6, whose axons project to subcortical targets. The next neurons to be born are the local interneurons in layer 4 of the cortex. Finally, the pyramidal cells of the upper layers 2 and 3 are generated. They send axons to other cortical areas. In other words, as cortical genesis persists, newly generated neurons migrate radially past the ones generated earlier to progressively create the 6 cortical layers in which neurons are located, according to their birth date, in an inside-out fashion. Early generated neurons thus reside in deep layers and late generated neurons in the upper layers. To sum up, the layered structure of the mature cerebral cortex is formed in an established chronological order. Thus, the so-called nature's most exquisite masterpiece, the layers of the cortex, are created in an inside-out order. Thanks for watching.